All right, so we'll get started. Welcome, everybody. Good morning. Thank you for joining us today for Forestry Friday webinar on Beech Leaf Disease. We're very fortunate today to have Cameron McIntire with us from the U.S. Forest Service Durham Field Office to talk to us about the status of Beech Leaf Disease in the Northeast U.S. Cameron is a plant pathologist with the U.S. Forest Service Division of State and Private Forestry based in Durham, New Hampshire. Cameron has a bachelor's in biology from the University of Utah and a PhD in natural resources and environmental science from UNH. His background is in ecosystem physiology, and he's been studying foliar pathogens of white pine and the hydraulic functioning of trees in response to drought. Cameron has been helping organize the federal survey and monitoring projects related to beech disease in New England since 2020. Couple housekeeping things before we get started. I'd ask you all to please remain muted throughout the presentation, and I'd also ask you to turn off your cameras during the presentation to preserve bandwidth and also to minimize distractions during the presentation. Also, please hold all your questions to the end of the presentation, and with that, unless there's anything else, let's go ahead and get started so there's plenty of time for questions at the end.
Okay, thanks for that intro, Aaron, and thanks for inviting me. I'm glad to have the opportunity to share some information about this new disease, especially with folks who are on the ground in an area where it's currently starting to establish and become a bit of an issue. So I'm going to start with a little bit of basic biology, since nematodes are not really something that I think most of us are associated with in a forest setting. They tend to be more common pests in an agricultural setting. But we're confident enough to say now that beech leaf disease is most likely caused by an invasive species of nematode. This is somewhat uncommon in the world of forest health. The only other major nematode pest of trees that I'm aware of is the pinewood nematode that can affect Scotch pine in parts of the eastern U.S. And so I wanted to start just by brushing up on some of the basic facts here. And despite a lot of shorthand referring to nematodes as worms, they do in fact exist in their own distinct clade apart from your traditional annelid worms or your earthworms and your leeches. In fact, they're more closely related to arthropods, and this is the group that includes many of our traditional forest pests like your wood-boring insects, your phloem feeders, ticks, and things of that nature. And they're thought to have split from insects evolving about 500 million years ago, with about 100 million years of separation between our earthworms. And the word the nematode is derived from the Greek word for thread, which is a pretty apt description because these are very thin and translucent. And because of that, it's very difficult to distinguish between species. They're very small. We measure them on the order of nanometers. On average, they're about 0.1 to 2.5 nanometers long. So not something that you're going to be able to see readily with your naked eye or perhaps even a hand lens. They can both be parasitic and free-living. They're parasitic on both plants and animals, like there are many nematodes that are probably in your gut right now that are part of the digestive process. Many of them are involved in decomposition. In fact, most of the nematodes that we're aware of inhabit about the top 10 centimeters of topsoil. And we believe there to be probably over 1 million species globally, but not even close to that many is yet to be described by science. So there's a lot of diversity out there. And the nematode that's associated with beech leaf disease, or BLD for short, is called Littlencus crenate mccannii. And the mccannii part is actually denoting a subspecies. And so this particular nematode is actually a subspecies of a recently described Japanese nematode, though it's still unclear if this was actually introduced from Japan or somewhere else. But it's probably likely from Asia. And it's important to mention that all trees with BLD have the nematode present, but there's still research underway to determine if they're a direct cause, either from their feeding or proteins that they secrete, or if it's possible that perhaps there's a bacteria or a virus that could be associated with them. And so nematodes themselves tend to be dimorphic, meaning that they exist with distinct male and female forms that can sexually reproduce. Here you can see the male variety on the top left, and the top right shows a corresponding female with her eggs here in this bottom left panel. And the only way that I can tell the difference from a male is they do have this little spike on one end of their body. And the juvenile down here is somewhat less distinct. The main difference for me is that it's much smaller and occurs, more towards like the middle of the summer, whereas the adults are what you're going to find in the late summer going into the winter. And you can see that the scale bars on here, they're on the order of nanometers. So it'd be nearly impossible to view these with the naked eye. And in fact, here's an example looking at a bud. And the buds of beech are actually where these overwinter. And so if you draw your eyes to this black box, you can kind of see this string right here. That's not a nematode. That's just like a fiber that's part of the bud. But if we continue to move in across these panels, so this is that same area, looking at a higher magnification, and if you move all the way in, we finally get to looking at this nematode that's kind of existing on the inside of this bud. And you can see this, as I mentioned, this very thin, translucent, worm-like animal. And here's what they actually look like in action. And this is a clip that was cut from a video that was prepared by the University of Maine Cooperative Forestry Research Unit. And I believe this is from a leaf that was collected at the Penobscot Experimental Forest. And this is how I actually do the majority of my diagnostics with this disease later in the season. If you think that you've found an infected leaf, you can simply cut it into pieces and place it into a petri dish with water for a few hours or maybe even overnight. And then if you stick it under a microscope, just a regular dissecting microscope with a backlight, you should be able to see what we call the swimmers, which is these guys that are wiggling around in the video. It's unlikely that these would be any other species of nematode other than that Litholinchus species. And when we do detections that are new to an area at the county level, we do like to have these confirmed by DNA sequencing. We have developed primers specific for detecting this species of nematode, and there are now several labs set up to handle that diagnostic. And I briefly want to talk about what we know in terms of the mechanistic impacts of this pest. And here you can see two micrographs of leaf surfaces. On the left shows a healthy leaf, and we see a diseased leaf on the right here. And these red arrows are pointing to the stomates, and this is where gas exchange occurs, and this is like the area of the leaf that's responsible for regulating photosynthesis. And on the photo to the left, we can see normal functioning stomates here. They're open, they're round, but on the diseased leaf on the right, it's much harder to make out these stomates. You can see one here that's sort of collapsed, similar here and down here with the other arrows. It's much harder to make them out. They're extremely deformed, and in some cases have probably lost function. And the slender lines that you see kind of going along the leaf here, these are actual nematodes that are just free living, existing on the outside of the leaf surface. But it's not just the outside of the leaf that's impacted here. In fact, most of the action appears to be happening from the inside. And here we're just looking at a beech leaf in cross section. And you can see all the hallmarks of a healthy leaf. You have the two different types of mesophyll on the top and the bottom plane. And then you're bounded by that waxy cuticle with stomates on the underside of the leaf. But let's check out an infected leaf. And of course, this is artificially colored here. And the nematodes are actually what's in blue. And clearly they're in a very high density inside the leaf. We're talking about thousands or tens of thousands of nematodes in an individual leaf. And it's clear to see that there's a lot of dysfunction happening here. The basic structures are breaking down, and these pests are feeding on parts of the leaf tissue. So this should give you some indication to just the mechanical havoc that this pest can cause. And now I want to talk a little bit about symptomology so we all know what to look for when we're out in the field. The photos I'm highlighting are what we refer to as the mild and really the most common symptoms of what we've been referring to as banding with this disease. And so what happens is that the infective leaves, they're actually a normal size and shape, but you can see that there's this dark band that occurs between the leaf veins, right? Right here, these dark bands here in this photo. And the number of these interveinal bands can be variable, but they typically cover less than 2/3 of the leaf surface, but there's really no pattern to the way they present themselves. If you just look at the smattering in this photo looking up on a canopy, you can see there's no real discernible pattern. Sometimes they're separated by a couple healthy bands. Sometimes there's a bunch of infected bands right on top of each other. You know, you might have 10 bands in one leaf and then maybe just 2 like you see here in this leaf. And I just want to mention, it makes a huge difference to look at these leaves with some backlighting. It can really help make this symptom pop out to the eyes. If someone were to send me the photo of maybe a sapling pointing towards the ground, it'd be much more difficult to distinguish this particular symptom. And the second more severe symptom is what we refer to as a more generalized leaf crinkling. And these leaves tend to be smaller than the normal size leaf, and they express these curled and shriveled edges right along the leaf margin. And they also become more thickened and a little bit leathery in texture. And these symptoms tend to express over the majority of the leaf as opposed to just a few bands. You can see here, this is a more advanced stage here where, you know, this almost looks like the leaves are kind of drying out and getting ready to fall off. And the other thing I want to mention is that this disease can also infect European beech, which is what you're looking at here in the bottom photo. But for most practical purposes, what we see in the field in a forestry setting, we're going to be dealing with American beech, although European beech is very common landscape tree that many people might have in their yards or in city streets. And then I just want to take a second to highlight some other leaf issues that are distinctly not beech leaf disease. Last year was actually my first time working with this disease in the field, and these are the most common things that I would find myself sort of doing double takes on. And by and large, the most common thing that gets mistaken for BLD is the rolling aphid. And this can seem to express leaf damage in a lot of different ways, but one of the symptoms of this pest feeding is the rolling of the leaf. You can kind of see it's like curling along the inside edge here. And this can look very similar to that crinkle symptom of BLD, but without the change in like the leathery texture that we see. In fact, these leaves tend to be sort of delicate, so it's just the opposite. They're sort of thin. And oftentimes, if you're not sure, if you were to flip one of these leaves over and look at the underside, you're more than likely to find little white aphids underneath. And then briefly, I mean, there is another aphid that affects beech, the beech blight aphid. These sort of dance around mostly on the stems, but sometimes you do find them hanging out in the leaf. They don't really cause a lot of damage to the leaf. One thing that looks like it causes a lot of damage, but is sort of superficial, is this ernium mite. It causes these patches here. It's sort of like a gall. And then something that can present in a lot of different ways is nutrient deficiencies here, which, you know, presumably this could be a nitrogen or a phosphorus deficiency, which can cause some discoloration. There's been some recent work tracking the phenology of this disease, and it's shown that symptoms don't really progress over the course of a single growing season. And that's just to say that when a leaf expands at bud break, the symptoms don't get worse. If it comes out with 5 bands, then those 5 bands persist and they don't increase in number later in the year. And we also don't see banded leaves, you know, progressing and becoming a crinkled leaf later in the growing season either. The one exception is that among leaves that show this crinkled symptomology, where in some cases those leaves actually become chlorotic and they will abort and drop off. And so there's some evidence to show that these aborted leaves will reflush and put out new leaves, but we don't really know very much yet about the conditions that have to be present for that to happen. I would imagine a lot of that probably depends on just the carbohydrate reserves in the tree and how much energy it feels like it can expend for that. We do see that the presentation of symptoms tend to stabilize over the course of 2 to 3 years after initial infection. And here we're looking at some of the results of some long-term monitoring data around Ohio, where the proportion of both banded and crinkled leaves are shown here, in addition to the asymptomatic leaves on this figure on the left. And we see that infected individuals exhibit about 25 to 30% of their crowns exhibiting symptoms on average, right? So it looks like maybe 25% remain banded even 4 or 5 years after infection. If you just look at the the solid black line of these box plots is going to be your average. And it's similar is true for the crinkled leaves. So even in trees that are diseased, most of the leaves actually seem to remain healthy or asymptomatic. It's not to say that there's not nematodes in there, but they just don't seem to be presenting symptoms. But the big caveat here is what we also observe is that these trees that are chronically infected, presenting symptoms year after year, they do have significant reductions in their total leaf cover. And then this corresponds with an increase in the proportion of dead branches, which is being shown here on the right. So you get here, we're just taking visual estimates of leaf cover, looking at leaf transparency. And so you can see a decline, especially following maybe the 3rd year of infection. It seems like you start to see a big drop off. You're starting to see more defoliation. And then, of course, the inverse associated with this is, once a branch has lost all its leaves, you start to see branch dieback as well. And then up to the point where you actually start to see tree mortality. And we have documented mortality attributed to BLD, both among mature and sapling size classes. It's not rapid. It follows multiple years of crown deterioration. And it's also likely that there are other stress factors that are associated with it. This could be things related to climate, perhaps drought, or it could be site conditions as well. These could be trees that are perhaps already growing on a poor site for beech habitat. And that might be what pulls the trigger for mortality in these cases. So as of fall 2021, this is where we stand. This is our most current map, but I will tell you that just over the last couple months, this is already outdated. But as of this past growing season, this is where our knowledge stands with the spread of this disease. It was first detected in Lake County, Ohio, in 2012, just right here on Lake Erie. So this is this is a popular port of entry right here. So not surprising to have an introduction of exotic pests in a place like this. There are currently 10 states in the U.S. that are affected, plus the province of Ontario, just north of the lake here. Last year, we had 47 new county level detections. That was the most that we've had since we started tracking in 2012. Last year was the first year that we got detections in Maine. I think this was sort of the big surprise for 2021. I believe Aaron already mentioned that he has an additional county here in Hancock for this year. Last year, we had no detections in Vermont or New Hampshire, which is sort of why the Maine explosion was really a surprise to us. But we actually did just get our first and only detection in New Hampshire last week, which is in the town of Derry, which is sort of in the southern part of the state, right around here where I've got my pointer now. And just one thing I want to mention about interpreting these sort of detections maps is that it can be, you need to be mindful of the size of some of these counties. It varies a lot across the country and within states. And so, for example, if you look at, let's see, what is this? This is Penobscot County up here in Maine, right? It's huge. We actually only had, I think, a couple detections in this whole area last year, and it was mostly on the southern part of the state. And you can fit, 14 Putnam County, New Yorks into that large area. And so this map is useful for documenting spread, but it doesn't account for the total area that we know to be affected by pathogens. So we know that this disease doesn't respect any sort of county line. So just take that into consideration when you're interpreting maps like this. as far as how this pest is moving around and spreading, we really don't know yet, and that's the most frustrating part of this disease. The mechanism of dispersal is still unknown, and we can't discount the possibility of multiple independent introductions either. You can see that there's considerable distance between some of the county level detections on this map, especially with what we observed in Maine last year. We know that we've seen BLD on European beech nursery stock, but one of the prevailing theories is that birds may actually play a role in the spread, which would explain some of these large jumps we've seen. But that's a very active area of research, and it's still being explored. So nothing conclusive there about how it's moving around. It's very possible that humans play a role in it. I don't think it's likely that it's not something that's going to be moved around in firewood like your emerald ash borer would be. We don't think that this nematode can exist in the vascular system, so it's very unlikely that it would be in the wood, but it's certainly possible that if you're working in a disease stand, a human could possibly transmit it from tree to tree if you're not careful. So I do encourage folks, if you're working in stands that you suspect have the disease, do the best to disinfect your boots, your gloves, whatever tools you might have been working with. It doesn't hurt to do it. I want to talk a little bit about survey efforts. We have a collaboration going on with our state partners and just the broader forestry community. And I want to pitch this app that was actually created for the sole purpose of tracking and reporting beech leaf disease. It's called the Tree Health Survey, and if you just search that on your phone, it should come up right in the App Store. It's free to download. And what this does is it allows users to upload location information and photos of beech trees pretty much wherever you have cell phone service. And this has been extremely useful for building our regional survey network. And at this stage, even asymptomatic information is valuable data to have. So I would encourage folks to check this out and contribute throughout your time in the woods this year. There's also a website where you can access this on your desktop. And you can create an account and not only input your survey points, but you can also view information from entries that other people have submitted too. And here I've just pulled up the information for I believe was our initial detection that we found in Lincolnville, Maine last summer. And so this includes, more information than just like presence absence data. You can also look at notes that people may have left with this disease and different metrics associated with how much of the crown was infected and some of the photos associated with it as well. And since 2019, there's been a push to develop a network of long-term monitoring plots for this disease. And this has been one of the main focuses of my work over the last year. Currently, we have a total of 151 plots established throughout the eastern US. And this map is showing the status of beech leaf disease in each of those plots as of last season. And most of these plots are going to be remeasured annually, but if a plot on this map has been marked as being unknown status, this sort of orange color, that just means that it was found to be negative for BLD in either 2019 or 2020, and we just haven't had a chance to remeasure it yet. So there's a few of those in Pennsylvania and Ohio, for example. These are the two states where it's actually been the longest, so we have a little bit more data here. But for this audience, I'm gonna share and focus mostly on some preliminary results of this monitoring with respect to the New England region and New York. And here we have a total of 65 plots so far. New York was the first state with a positive detection, and they began establishing plots in 2019, with Connecticut following in 2020. And then every other plot in New England was established just this past year in 2021. So this was a huge effort by Aaron and our other forest health partners, and something that I'm really grateful for. And just to give a little overview of what we're measuring these plots, we're using a fixed area 10th acre plot with seedling and sapling subplots. But for this talk, I'm going to be mostly focusing on what we're observing with mature trees. And really, among the goals of this work is to quantify some of the site attributes and the stand attributes that may play a role in either the spread or severity of this disease, but also to track the progression of it over time. That's really the the main bonus of having a long-term monitoring plot network. And so just as an example, some of the site attributes we may be interested in are site level topography and climate. Stand attributes include things like stem density and stalking, tree size and crown class, species composition. Like we're certainly going to be interested in, how much beech plays a role in this disease. And then we have some more quantitative metrics where we're taking observations of beech leaf disease and also the bark disease in these stands and making note of crown density and dieback that we observe. And so here's just an example of some of the site level metrics that we were able to grab from these plots right here. You'll see there's a large distribution across all of these metrics. And with a monitoring plot like this, variability is a very good thing, right? We want to have a bunch of different sources to grab from so that we can do analyses down the road and start to try to pin specific factors onto, where we see more decline, like where do we see this disease more severe, or even the other end of the spectrum. It's like, where are the stands that seem to be resisting this disease in the long term? So you can see like we have, a broad distribution in elevation and slope. And among those plots that are on slopes, we have a pretty good distribution in the aspect that those plots are facing in. And then we have more of our stand structural qualities here, like we have our distribution in trees per acre, our basal area, and then our tree size as well. We have a total of 28 species that were represented in the Overstory. By and large, beech is the dominant here, and that's sort of by design, right? These plots aren't meant to be like random FIA plots or anything like that. Like we're looking specifically for this disease, so we've targeted stands that are heavy to beech. But the other dominant components that we've seen throughout this part of the country is red oak and white pine are the top two. But there's also a pretty strong component of red maple, sugar maple, and hemlock, in addition to a whole bunch of species that are a minor component. And keep in mind, these are just overstory trees here. So these are mature trees greater than 5 centimeters, or 5 inches rather. And then foresters in the audience should be more familiar with, I think, some of these stalking indices here. So here I've plotted Reinecke's stand density index as a function of our stand's mean diameter. And this is really, again, just to show that we have a lot of variability in the stalking density of these stands, which I think is going to be really powerful for some of these analyses down the road. So I want to share a little bit of what we found with respect to beech leaf disease in these plots last year. And among the mature trees, I've broken down our data into different crown classes. And so what you're looking at on the x-axis of these graphs are the codominant, which is CD, so codominant, intermediate, and suppressed crown classes. And this here shows all the trees that we measured to be positive for beech leaf disease last year. And so that ended up being just over 500 trees. 38% of those were codominant, 28% were intermediate, and then almost half of the total were suppressed trees. So these are trees that are sort of below the general canopy line. So not really quite in the understory, but mature trees greater than 5 inches in diameter. And within the canopy, we found that trends were very similar to that figure I showed you earlier in Ohio, where the disease has been established for some time now. And among all trees in all the size classes, the majority of the leaves are still showing no symptoms. What I've coded here is normal or healthy leaves. But if you look at sort of the progression through the crown here, on average, about 60% of those trees are asymptomatic, but about 20 to 30% express that striping or that banding symptom. And a much lower proportion, about 10 to 20%, express that more severe symptom of that shrinking and that curling and that changing in texture. It's possible that we could be missing some of this more severe symptom, the crinkling, because as I mentioned, some of those leaves can fall off. So it's hard for us to measure things that aren't there. But that definitely gets accounted for in our measurements of crown dieback. And so here's what we're what we found in terms of crown density and dieback. And in this grouping here, I've actually grouped our dominant and co-dominant trees in black, and then our intermediate and suppressed trees in gray here. And we're looking at just beech trees here. So FAGR, that's just a species code that stands for Fagus grandifolia, which is the species for American beech. So this is actually our healthy trees here. And then these are our BLD infected trees. So across all the plots, and this is over 1,000 trees, we don't see any clear differences in crown density, even in comparison with all other trees. So this is grouping in our oaks and our maples and everything else. And the reason that we measure the oaks and the maples is because we want to know if there's another pest in there that could be causing defoliation that might confound some of our measurements. For example, if we're in an area where we're having a spongy moth or maybe a brown tail outbreak, we want to know if the defoliation that we're seeing, is that actually caused by BLD, or is that something that's affecting kind of the forest at large? But last year, I don't think that was really an issue in any of our plots. So we found actually really similar metrics of crown density across the board. But if we subset our data and only look at our disease plots, we do see some indication of decline becoming evident. And this is especially true in areas where the disease has been established for a little longer. Places like New York, for example, where it's been for a few years, you see crown density here is actually much lower in some of these blue bars than it is in places like Maine and Connecticut, where it's relatively new. That's not to say that we didn't see some pretty bad infections last year, but on average, it's still pretty new to the region. So we may expect this to progress and get worse over time. And looking at crown dieback, it does appear that overall, the intermediate and suppressed trees are exhibiting higher levels of dieback among those trees that are affected with BLD. It's about an order of magnitude greater amount of dieback in beech trees that are infected with BLD than what we see in the healthy trees. And so this is an interesting finding. Suppressed trees tend to have a little bit higher level of competition, so that it might be that additional level of stress that could be pushing this disease to progress and deteriorate the crown a little bit faster. And again, we can break out this at the plot level and see that there's a lot more variability here, which probably relates a little bit to some of the structural complexity in these stands. And I also want to mention, we can't really talk about beech without acknowledging beech bark disease. And this is something that we've also measured in these plots. We measure the severity of both the scale insect and the cankering on all of our trees. And what's unique about the monitoring plots in New England is that we are very much entrenched in this disease, right? It's been moving through for almost 100 years now, and many of our plots are in what we call sort of that aftermath of forest, especially much of Maine. And then you find in Maine, this is just looking at presence-absence data, but nearly 100% of our trees are afflicted by either the scale or the canker or both. But in addition to just the presence absence, we also take a more relative metric of severity of both the scale and the canker. And we do this on sort of a categorical scale. We do it visually. We're not out there like counting scales or measuring the size of cankers or anything like that. But we do rate it on sort of a scale from, trace to light to moderate to heavy, right? And I think once you have a trained eye out in the field doing that, I feel pretty good about the data that we have coming in from our state cooperators on this. And again, it's not a surprise here in states like Maine, we see a much higher incidence and severity of the canker. That's true for Maine, it's true for New Hampshire and Vermont as well. So these are areas where, we now have potentially two diseases affecting different parts of the tree. So one of the things that I'm interested in pursuing and how these interactions between the two diseases is going to play a role in the health of and the overall health of beech moving forward. And here, what I've done, I've just set up some matrices corresponding to beech leaf disease on the x-axis here, so sort of showing a progression from a healthy leaf to a diseased leaf, and then doing pretty much the same thing on the y-axis with beech bark disease, looking at the trees that we find with this clean, smooth bark. These are probably trees that, if you're finding them in Maine, they probably have an innate genetic resistance to the disease. We know that this is something that exists. It's very rare. It's probably only about 1% of the trees on the landscape. But then we have this progression to what I think most of us are more common or accustomed to seeing on beech, which is this heavy cankering. We don't really know anything about resistant to beech leaf disease yet. It seems very possible. I've seen tree or stands that are hammered, and then I'll find individual trees in there that seem like they're showing some level of resistance, but I think it's a little bit too early in the spread of this disease to know for sure. And that's really where the strength of the monitoring plots are going to come in, is to really evaluate that over time. But just to look at this matrix here, I mean, basically what we can do is we can start pulling out some of our response variables and just looking at where those lie and try to pull out meaningful information. So, you might expect there to be some association with DBH, tree diameter, in terms of the severity that we see in coordination with both the leaf disease and the bark disease. We might be able to say the same thing about crown density and crown dieback. The areas that are coded red in these plots are metrics that are more intense. The damage is more severe. But I should note here that the majority of the trees that we sample fall within this space, right? This is sort of like the normal distribution of our data. Because we might only have a handful of replicates that fall into the extreme either ends of these categories. Like, for example, if you look at the top right corner here, this would be a tree that rates very high for beech leaf disease, and then also receives the highest rating for the bark disease. And we didn't measure any trees in that instance. So most of the things, most of the damage we see falls kind of in this space. So I think it's going to be, really important to assess the changes in time as this disease progresses. Right now we just have access to 1 year of data in New England. So we're going to have to be, vigilant and continue to monitor this disease over the next few years. Based on the data that we see coming out of Ohio and Pennsylvania and New York, it doesn't seem like this disease is going to be something that just sort of goes away on its own. And I also just want to share a few resources that are out there for folks. Cleveland Metro Parks, this is the group that actually first discovered the disease in Ohio, and they've really spearheaded not just a lot of the research, but they've assembled and created a lot of great fact sheets and publications for folks that are interested in this disease. So if you're interested in this, or if you think that you may have BLD, on your land, this is a great resource to access to. I'll also mention that the Forest Service published a pest alert last year on that. I have tons of these at our office, so if anyone wants some of these, I can mail you some copies. There's also a PDF version of this online that I can link you to. And then for folks that really want to do a deep dive, there's a lot of peer-reviewed research that's been coming out just in the last 2 years. I think a lot of this is going to be open source, but if anyone has trouble accessing any of this and they want to read some of this stuff, just let me know and I can make sure I get that to you. And I also just want to acknowledge, there's an amazing group of partners that have really spearheaded the work around the disease since its initial discovery. And all these folks have contributed to aspects of identification and research and these survey and monitoring efforts throughout the eastern U.S. So it's worth noting that I'm pretty new to working with this disease. And so I'm really here just to help spread the word and share kind of the foundation of this knowledge. And, that's actually it for my portion of the talk. So I was going to hand it over to Aaron now, and he was going to talk a little bit more specifically about what's going on in Maine. And then I think after Aaron, we should have some time for questions.
Yeah, thanks, Karen. And I'll just continue with your talk about partners. I think the same can be said for the situation with beech leaf disease here in Maine. We're a pretty small outfit, the insect and disease folks here, and the reason we know so much about where beech leaf disease is in Maine is because of partnerships, other organizations, other departments within the state. Landowners have been critical in alerting us to locations. It's been I think one of the a silver lining to the whole beech leaf disease story in Maine has been kind of the way that people have been informed and have reported new locations and have been interested and very free with sharing information. So if there is, like I said, to be a silver lining with beech leaf disease, I would say that's it, and I hope that continues, and it does continue. I mean, I get reports about beech leaf disease on a daily basis, several of them from various cooperators and new cooperators all the time. So really hoping that that's gonna continue. So just to quickly talk about the situation here in Maine. Back in 2021, we saw a small number of very severely affected areas in Knox and Waldo County. As we were first alerted to this by a landowner who was very connected to their land and their beech trees, their beloved beech trees. Hadn't noticed anything the year before, and then in 2021, saw some pretty advanced and severe symptoms in their trees. Alerted myself. I then relayed the information to Cameron. Cameron came up. We took a look and saw that there was very likely beech leaf disease. It was confirmed via PCR by Bob Mara and in Connecticut. But these very severely or fairly heavily impacted stands were not very numerous. We found several of them in Knox and Waldo County, but not much outside of that. Once we did start to do more of a directed survey and got some people from our technicians and some of the other professionals that worked with the Maine Forest Service, really got out and started looking, and also Department of Agriculture was instrumental in helping us with that. We started to find many more trace infestations. So you see in the disease distribution figure here that 2021 detections are in purple, and that's mostly trace infections, trace impacts and stands. Then now in 2022, we're not very deep into 2022. We found a lot more trace infections, pretty far-reaching from the initial finds in the state. Again, facilitated by partners, and we found a lot more trace impacts far away from those initial Knox and Waldo County detections, and within Knox and Waldo County, we found a lot more very heavily impacted stands, and I'll show a picture of that a little bit later here. So to date, the furthest west location in Maine is in Somerville in Lincoln County. The furthest east is Ellsworth in Hancock County. The furthest north is just south of Burlington, Maine in Penobscot County. And it goes right to the coast to the south. So that's and it's been found in on Islesboro. So it crosses the water quite easily. It seems. If you could advance the slide, please, Cameron. Yeah, so this is just a closer look of the locations to date, and this is expected to expand as we get more people out in the field. We've done some trainings this year, and I think the more informed people that are out on the landscape and paying attention to beech, we're going to find perhaps that this is present even further from the boundaries that are presented here. And if you could advance the slide, please. So this is what I call ground zero. This is the location where beech leaf disease was first reported. And this is May 29th in 2021. As you can see, this stand is pretty heavily impacted by beech bark disease, but beech leaf disease was pretty advanced at this time in late May, and you can see, well, you can't really see great detail on this, but you can see a variety of there's a variety of symptoms presented here, from banding to some asymptomatic leaves, and also the sort of more severe crinkling and leathery leaf type symptoms. But this was not only understory trees; it was already in the overstory. And again, the landowners that reported this, they have a small trail that goes through their woods that they may walk at least once a day, if not several times a day, to just make observations. Very observant people, very in tune with their land, and they hadn't noticed anything the year before. And then at leaf out in May 2021, this is the reality that they faced. I believe the next slide, if you could advance it, is this year. So this was May 16th. People will say that phenology was a little bit ahead in Maine in 2022. So this is not a great comparison, but you can see. I mean, this is after bud break, and you can see that things are very, very different. Um, le- these trees just were not leafing out, and there were very few leaves, and many of the leaves that were out had some pretty severe symptoms. I just got a text with a picture. I think it was yesterday, from these landowners, and some of these trees have set new buds and pushed out new leaves. I don't know the extent to which that's happened, but that was interesting to hear, and I'll be following up on that with the landowners here in the coming weeks to see how this is progressing. But this was a bit shocking to me when I came to see this stand 1 year after the initial detection, or the initial report and confirmation of beech leaf disease in Maine. The next slide, please. So one really recent development has been a treatment option, and this is based on trials that were done in Ohio by the Cleveland Metro Parks people, in partnership with some of the larger tree care companies in that area. They had been trialing several different treatments and modes of delivering those treatments since 2017. And they found that the most promising treatment has been a high potassium fertilizer. These are generally referred to as phosphite products. The product they used in their trial was called Phosphite 30, which is like I said, a high potassium fertilizer with a NPK ratio of 0.027. And the general recipe is for every inch of diameter at breast height, which many of you know is taken at a specific level of the tree. You can either take circumference and turn it into diameter, or just use a diameter tape. And for every inch of diameter, 2 ounces of polyphosphate 30 were diluted in 14 ounces of water, and that final solution was applied to the root zone of trees. And that can be done, or that has been done, either as a soil drench, as you can see in the picture, or as a soil injection, and that of course requires soil injection equipment. In order to do this, you remove leaf debris, any kind of anything above the soil, and to expose the soil to enhance absorption of the product, and then that product is dumped in the root zone. If the soil is not really absorbing the product well, you can dig a trench, just making sure to, or if the tree's on a slope, you may need to dig a trench. If you're doing the drench rather than the injection. Anyway, it's important that the product gets into the soil and doesn't run off, because that's where it's going to be absorbed by the roots, brought into the tree, and then do whatever it does. I don't know exactly the mechanism of how this works. I'm not sure anybody does, and unless Cameron has some information about that. But they have noticed that in smaller trees, so this trial was done on smaller trees, they found that it's really has a positive impact on canopy and canopy density and just overall crown health. I was talking to the person who in Cleveland with Cleveland Metro Parks who was heading up the trial, and he said that when you look at the control versus the treated trees with the phosphite 30, it's pretty remarkable the difference in tree health. So this is very, very new information. This is on the Maine Forest Service Beech Leaf Disease website. It was just put up just very recently, just a day or two ago. And again, this is the best information we have. We wanted to get this out to, it's not necessarily applicable to a forest setting, but for those people who have beech trees in the horticultural setting as elements of their landscape, their yard, this could be an option to try. Again, this is done. The trial was done for smaller trees, so the impact of this treatment on larger trees is still sort of unclear, but it is the only treatment that has been trialed and has been shown to work fairly well. So this information is out there now, and encourage you to look at it if this is something you want to consider for some of your trees. That's pretty much all I had today to talk about. I just wanted to provide a little update, more Maine specific information, because I know a lot of people are wondering about exactly what beech leaf disease is doing in Maine, how fast it's spreading, or where it's currently found. So with that, I will go back on mute, and we'll start to take some questions. I don't know if anybody's been monitoring the chat. I haven't been able to do that yet, but I'll take a look at that now. If people want to ask questions, of course you can raise your hand, and we'll address the questions one by one. I'll kind of field questions for folks, for people's questions for Cameron. We've got a question from Nancy Olmstead. Go ahead with your question, Nancy.
Hi, yes, good morning. Thanks so much for this great webinar. Cameron, I wondered if you could help me backfill my knowledge a little bit. You showed a slide about beech bark disease severity and incidence, and showing that that's worse in Maine and New Hampshire than in other New England states. And I was just wondering, do we have an understanding of why that is, or does that have to do with the history of the disease? I was just struck by that and wondered if you could say a few words.
Sure, yeah, that's a great question, Nancy. So beech bark disease was first introduced from Nova Scotia, and unlike the leaf disease, it has like a very distinct, what we call an advancing front. So there's sort of a leading edge of infection followed by mortality that progressively spreads. And so it's been spreading from that origin in Nova Scotia. And of course, the first place that it hits was Maine, and then moved through New Hampshire and Vermont, and is now that aftermath region is starting to make its way, or I shouldn't say aftermath, but because aftermath refers to really forests that have already experienced the disease and are starting to have mortality. But that advancing front is very much now moving through places like Connecticut and New York, and just really starting to get in areas like Pennsylvania. And so areas where there's an interesting intersection between where the leaf disease has been for a while versus where the bark disease has been for a while. And I think that interface is really starting to collide in the last couple of years.
All right, we've got a question from Sue Hatch. Sue, go ahead, please, with your question.
Oh.
Sue, do you still have a question? If not, we'll go to.
Can you hear me now?
Yes, can hear you now. Go ahead.
Yes. Okay, great. I have a lot of questions. I'll try to go through them here and maybe just ask them and then you can answer them, or do you want me to do them one by one? Does it matter?
I think probably because for the sake of my memory, let's do one at a time.
When did these nematodes hatch from eggs? What time of year?
That's a great question. They would be hatching probably in the next couple months. We know right now if we try to extract them from leaves, you're not going to find any of those swimmers. We don't start to see the adult nematodes swimming until later in either late July or August.
So the damage to the buds is from the eggs? What's the damage? No, all my trees have, I mean, not every bud, but many buds dead this spring. I assume that's damage from the prior year, or, I mean, the buds form in the fall.
Yeah, we believe a lot of the damage is occurring inside the bud.
Okay.
They come leafed out with symptoms already present.
Yes, definitely. I have a aphid that has white fluff on it, some kind of woolly aphid, as a co-infection on this is an ornamental European beech. I also have tons of regular beeches around. And I also have tiny caterpillars showing up. Now these trees have, blessed their hearts, lost almost, tried to leaf out, had crinkling, banding, everything. And now they're putting out new leaves that look pretty good, but they've got these aphids and caterpillars on them. So that, just telling you that, if you have anything I should do about it, I don't know if I should try to treat the aphids with, a permethrin or neem or something like that.
Yeah, we see co-infections are very common. It's not a surprise to see 3 or 4 different pests associated with these leaves. I think most of those other pests are pretty superficial on beech. You can certainly treat if you want. I think a lot of that damage we consider to be more aesthetic than really extremely harmful to the tree.
Okay, well, that's good to know. Are the new leaves going to be infected? The ones that are just coming out now, are they going to get infected too? Or no, because the activity has diminished, or what?
From the little data that we have on reflushing, it can be either or. We see that even in a given year in a disease stand, still a lot of leaves flush out as asymptomatic. So it's really anyone's guess whether or not the nematodes had a chance to get into those buds and do the damage.
Okay. And that treatment, which I'm thrilled to hear about, when, like right now, and repeated every month? Should it be started in May? When should it start?
As far as I know, I believe the treatment can happen anytime during leaf out. It does need to be, uptake needs to occur because we're not doing an injection. So there needs to be transpiration occurring. Aaron has been working more closely with that, so you can correct me if I'm wrong, if there's a more ideal time during the growing season.
Yeah, I left out some critical details when I was talking about that. I apologize for that. So the treatment should be done sometime in June, preferably a little bit earlier in June, but now is a good time. And then again, about a month to 8 weeks after that, a second treatment. And the recommendation is to continue that treatment for 5 or at least 5 years. So that's the kind of the treatment window. But I think there's definitely some wiggle room there. But I think the sooner the better is kind of the conventional thought on that.
Okay, well, that's good. Well, thank you. That's the first round of my questions. So carry on, everybody.
All right, thank you. So we have a question next up is Mike Parisio with Maine Forest Service. Go ahead with your question, Mike.
Hey, thanks to you both. Cameron, just curious, the most interesting aspect of this system, is one of the unknowns, the mode of spread or whatever. So I know nobody knows what's going on yet, but I'm just curious. I'd love to hear about any, weird vector hypotheses or what people are actually digging into. I just find that interesting. So any insight on perspective vectors, that's developed in, this rapidly developing story.
Yeah, great question. That's usually the question on the tip of everyone's mind. Allison, you can go ahead and turn the recording off while I speculate. No, I'm just kidding. So with the bird thing, so there's an active research project going on. There are several species of songbird that we know seem to actually prefer beech buds as a food source. And they've been doing trapping and collecting the birds and collecting feces to see if they can find any evidence of nematode DNA in there or even intact nematodes. And what they've been able to find so far, it is interesting. It does appear that both mature nematodes and eggs can survive the gut of a bird and end up in the feces. Of course, the feces can end up virtually anywhere. It's unclear at this point whether or not that's actually a viable mode of transmission. Like, no one has been able to like sort of connect the dots and like prove that eggs that have passed through the gut can actually hatch. And once they do hatch, like, how do they get into the bud? Do they have to crawl into the stomates? So there's still a lot of connections that we need to make. So that's just pure speculation, but it's sort of a leading theory because it helps explain why some of those detections we get are so disparate, unless there's some sort of human vector involved as well. I will say the other popular theories out there, it could be something as simple as being windblown. You know, these nematodes could just quest on the end of a leaf, and if they pick up a gust, they're so small and light, it's not unreasonable for them to travel easily within a crown or within a stand, but it's anyone's guess how far they could go. But I think I'll leave it at that for now.
Fascinating. Thank you.
All right, another question by Amy Campbell. Amy, go ahead with your question, please.
Can you hear me okay?
Yep.
Yeah.
Okay,
Yes.
great. In regards to the transmission, maybe this is just, happenstance, but we have a lake property in Camden on the Hope Line, and had quite a severe infection there last summer. And then we live in Rockport also, and noticed some effect also last summer, but much worse this year. So I'm wondering whether it's human transportation, on the tires, on cars, or whatever. Again, this could be totally just, happenstance or coincidence.
Yeah, it's totally possible. I mean, I think without a question, like, the initial introduction was probably through a trade port in Ohio. It's very likely that the epicenter in southeastern New York was a separate introduction. It seems likely that humans could have brought it there. But I kind of throw my hands up. It's like, yeah, humans could have moved it, but like, how? It's like, we don't know if it's coming on leaf material or nursery stock or anything else. I mean, some of the detections that we've seen pop up this year, there's just, there's no other beech trees around that are infected. It just seems to be in the middle of nowhere. So it's a frustrating thing at this stage to not know, because once we know, we could start to recommend different sort of quarantining efforts and just kind of spread the knowledge. But right now, I'm afraid I don't have much to offer in that regard.
Yeah.
Thank you.
Yeah. Thank you for your question. Cameron, I just wanted to throw this out. I got a request. People are starting to, they're starting to remove their trees, cut down their trees that are very severely impacted, and they've kind of lost hope for. And I'm starting to get questions about, what's safe to do. Can we move this material off site? And the recommendation I've been getting is, if you burn wood at home for home heating, keep the firewood there. If you need wood chips for landscaping, keep the material there. I'm just trying to encourage people to not move it, but I don't know what the level of risk is in moving, this woody material off site after it's cut down. Branches with leaves and so forth.
Mhm.
So I've just been encouraging people to move material as little as possible, and encourage those the tree care people to, you know, dispose of the material in a municipal landfill, and not have a you know, I know some tree companies have a dump site, you know, in some random place in the woods. So I've been just really trying to encourage people to keep it local until we know more and can really give some recommendations. But I don't know if you have any thoughts on that.
No, that's a great point, Aaron. Our recommendation is, if you have the facility to do it, burn it on site. I don't think it gets better than that. And we've been even making that recommendation. You know, we met with some of the nursery folks here in New Hampshire, because that detection that popped up here was just a lone European beech tree. And so we know that's something that's present at nurseries. And so nurseries are no stranger to dealing with pests and diseases, and so they usually have the ability to do it. But if you're a small landowner, that can be more difficult. But I would say, if you're able to not move it, don't move it. If you have to move it, maybe just let it dry out and desiccate in your yard for a year or something. That would make me feel a little bit better about it. But it's not a federally regulated pest at this point, and so it's not strictly illegal for you to be moving it. But it's really just, I think, as you mentioned, Aaron, some of the best practices there.
Yeah, thanks for your insight on that. Does anybody else have any more questions for Cameron, or perhaps for me, about beech leaf disease, or any of the details associated with beech leaf disease? Now's your chance. We're up, you know, we're over the hour, but we perhaps could take another question if anybody's got one.
Yeah, I'm happy to
Nancy?
hang out for a couple more minutes.
Nancy, did you have
Yeah.
another question?
Thanks. I just had one comment or question, which is, are you looking for more plot locations? I work for the Nature Conservancy now, and we certainly have tons of beech on our properties. So just let us know if, you know, you're looking for more plot locations.
Great. Yeah. Why don't you get in touch with me and Aaron about that?
Yeah, Nancy, feel free to reach out to me. I believe you've got my contact information. That's a nice offer. Appreciate it.
Yeah, sure thing. And I should say, I probably can't officially make the offer myself, but it occurred to me and we could talk about it. So thanks. Yep.
Thank you.
Okay, great. All right, if there's no more questions, thank you very much, Cameron, for joining us today to talk about current information on beech leaf disease. Really appreciate you taking the time to talk with us about that today. And I'll look forward to seeing you a little bit later on this summer when we do our long-term monitoring plots, and hopefully there's no surprises.
Cool. Thanks, Aaron. Thanks, Allison.
Okay. Okay, so that concludes our Forestry Friday webinar. The next webinar will be July 15th at 9 o'clock. Forest Protection Team will be covering Maine's wildfire weather program and predictive services for wildfire potential. So mark your calendars for that, and make sure to get registered for that and future Forestry Friday webinars. Again, thank you to everybody who participated and showed interest today. Enjoy the weekend.